and it's on stormwater toxicity to salmon, which of course, anything to do with salmon around here is interesting. So I'll talk about that as well. So non-targeted analysis. So what we normally do is targeted analysis. So whether we're looking at BTEX, a few compounds, or doing a Fisher list, we're using an approved method to identify a few to 100 plus compounds, and they're compounds that we come up with the list. We have standards. We know how to measure them. We know how to quantify them. And non-targeted, I use this picture of the sky with the big net because it's really a sort of a big net approach. It's a way to not come in, well, let's look for X. It's let's using these rapidly improving chemical methods, how can we identify as many compounds as possible? And I'll show examples of we're identifying 1,000 to 2,000 or more individual chemical analytes per sample. It's a hot topic. This is just some titles of some recent publications. I'm sort of nerdy. I start my mornings looking at environmental science and technology, what's new today. And if you look at the titles, we won't go over them, you'll see surface waters, wastewater treatment plants, sediments, all these different media that people are looking at. EPA, as we look at the lower kind of right-hand corner, they have folks down in Research Triangle looking at non-targeted analysis. If you look under the upper, CTAC, they have a conference, again in the Research Triangle, coming up in a couple months, getting all the folks working on this topic together and trying to see where we are. And just to kind of show how rapidly things are coming, this morning, I said, okay, what's new on ESNT that may be of interest to this audience? And here we have non-targeted analysis of streams. And I like this figure because it kind of shows the whole gamut of issues that we deal with, everything from sampling to analytical. And then when you do analytical, you end up with lots of data per sample. And so there's a lot of multivariate ways we need to evaluate that. In our world, there's probably, when I was looking at the slide that Dimitri put up at first, and it said, folks in this group work in these various medias, and we have them all. People are looking at surface water, storm water. I'll talk about some groundwater. I think since technology is going to have a lot of influence on wastewater treatment. And then people also in sediments, air, and dust. And then sort of outside our world, well, drinking water's in our world. People are looking at that. And then, it's easy. One of the messages is this fancy analytical equipment is getting cheaper. More universities are getting their hands on them. And, it's not that hard to go down to the store, buy a few things, sample them, and come up with, wow, look at all these contaminants or chemicals that we found that we didn't expect. So sort of what are the implications? Again, it's sort of the detection of unexpected contaminants and transformation products, which I'll talk a little bit more about in one of the actually both of the case studies. And so I think I have a fear or thought that, plaintiffs' attorneys may be going out and saying, oh, let's just analyze everything, see what we find. And these things are very sensitive, so there may be, sort of the ubiquitous background concentrations of chemicals that we kind of understand in our world. We may find more of them. There may be a little bit of everything everywhere. There's going to be identifications of new contaminants concerned. The second case study will sort of focus on that issue. It'll help us kind of understand fate and transport, as we're used targeted analysis. It's sort of, we have a narrow window on what's going on. And I think as we learn more, as we look at these issues broader with a broader chemical suite, we'll be able to understand them better. And we'll see some of that in the case studies. And then as Dimitri said, he and we like to do forensics. And I think the more chemicals we have per sample, the more likely we're going to be able to find chemical differences that allow better forensics and better source characterization. So there's a lot of non-targeted methods, but the sort of two big areas in the organic world is multidimensional chromatography and then increasingly higher resolution mass spectrometry. So we've all sent samples throughout our career for GC or maybe HPLC. Each of those separates contaminants using basically one factor. GC, it's usually boiling point. HPLC, it's polarity. But with multidimensional chromatography, you can add multiple steps of separation. So I work with GCXGC-MS, and I'll share you sort of what the inside looks like in a slide or two. But basically what it does, it sends the sample through one column, separating it by GC or by temperature like we normally do, and it takes little pieces of that sample coming off, puts it on the second column, and separates it by polarity. And so you pull those unseparated compounds and are allowed to separate them more. Wait a second, the slide's getting ahead of me. Okay, so and then mass spec or high resolution mass spec, what it does is we typically don't do separation before it, although you can use HPLC. It just is so good at noticing minor differences in the complete compound is it can come up with a chemical formulae. And so what we'll see is that we can identify thousands of formulae per environmental sample. We may not have the structural information, but we have information about a lot more of the materials that are in a sample. Here's the GC. As I said, I don't know if the pointer is working on the PowerPoint. I mean, from where you guys are seeing it. But column one is a typical GC column. It goes through modulator where it freezes a little bit every second or two as some of the material coming off separates it and puts it on another column. And so you get what have been, what looked like one peak in one dimension, we see as actually multiple compounds. And each one of those peaks then goes into a mass spec, and we can then compare them to libraries and come up with what we call tentatively identified compounds. That's kind of our best understanding of which each of those peaks are. The high-res mass spec, again, it typically just identifies at the formulae level, but what you see here is just 5 spectra from the first case study. But what I'm trying to show here is that just across that little period of time covered by the red dot here in C, we get this many formulae. So when if you looked across this entire run, I think we had 2,500 or more formulae that we were able to identify, that we were then able to understand the chemistry and compare samples across the site, understand how do these formulae, are they similar or different to ones we see in other types of samples. NTA should be used with caution. All these methods are still being developed. There's not a lot of certified NTA approaches that are in labs that the state has said, yeah, this is great. A lot of it is currently being done either at the academic or corporate labs. So each method's a little different. The figure on the right is from the EPA. They're doing a multiple lab study where they made a spike standard with 1,269 compounds, and they're just showing here, how many compounds were identified by each of these different methods. And sort of the spooky thing is like 37 were identified by all 3, and some weren't identified at all, and others others were identified by one or the others. And so we're still don't understand false negatives and positives in a lot of these methods. So they're improving, we're learning more, but people are using them more in places, but there's still evolution till they're going to be sort of something that's going to be we'll have at least we'll understand the levels of uncertainty. And with that, I'll just jump into the case studies. So this is a project that I've been involved with, working through Chevron or with chemists and toxicologists at Chevron since about 2010. And the question was really from the regulators, what is in samples after the groundwater after bacteria degrades the hydrocarbons? And so what we've been trying to do is identify and characterize, what these metabolites are, what are they, how toxic are they? And then over the last few years, originally we assumed everything we saw were breakdown products of hydrocarbons, but now we realize that some of them may be products of microbial biosynthesis, and I'll explain that more in a slide or two. So if you've worked at petroleum contaminated sites, you send groundwater out for analysis for TPH, or around here we call it the Northwest TPHd method. And what you often get back is sort of a hump in the diesel range, and you get the machine calibrates that area and says, oh yeah, that much hydrocarbons. But if you run that extract through silica gel and pull out everything, polar compounds, and let just the hydrocarbons come through, you see there's actually very low concentrations of hydrocarbons, but higher concentrations of this material that's pulled out by silica gel, which means it's chemically polar, and those are the compounds that we assumed are these bacterial metabolites. So what we've been doing is trying to understand what they are. And this is just sort of the current status in Washington on this issue, although DOE is, we've communicated and talked to them a lot about this. They're doing a lot of their own in-house toxicity evaluation, so I think they're going to come up with some new thoughts over the next year. But currently in Washington, we're not allowed to, we have to compare the non-silica gel treated sample to the regulatory limit based on hydrocarbons, even though we know in many cases that what we're detecting is not hydrocarbons. So this program that we've done, it's a combination of biochemistry, that's how I got involved. What metabolites do we expect from hydrocarbon breakdown? This non-targeted analysis to understand what do we actually see. The toxicologist then said, well, how toxic are these materials? And then we've put it all together. We've collected data at a lot of sites, over 100 sites, using these methods, and tried to come up with some ways to develop a regulatory framework. So for those of you who haven't taken hydrocarbon biodegradation in a while, we know the classes of compounds are produced kind of no matter what you start with. The bacteria add oxygens, and so we have alcohols, ketones, esters, alcohols, and acids. These are sort of oxygen-containing compounds. And if they're in the degradation pathway, they still have the carbon structure of the hydrocarbon, but they have some oxygens on them. And I think it's that carbon structure of the hydrocarbons that makes the toxicologists and regulators, ask questions. What we did, because there's hundreds or thousands of individual hydrocarbons in a gasoline or a diesel, we couldn't identify everything, but we know the basic chemical structures of the hydrocarbons. We know the type of oxygen-containing compounds that bacteria will produce from them. And so we sort of put together these 25 classes of compounds. The toxicologists came up with toxic values for them based on compounds that have been tested in those classes. And then we use the chemistry to identify, well, what fraction of the compounds we're identifying are in each of these classes, and what does that say about the toxicity of the complete mixture. And again, I'm going through this 10 years of stuff pretty quick, so if you have questions about this afterwards and you want to contact me, I can send you papers. We're happy to discuss it more on the phone. real simply, what we found looking at 100+ service stations and marketing sites is we get near the source zone where there's still hydrocarbons, we have more compounds earlier in the pathways, more of the ketones, maybe we see some phenols. But as you get down gradient, we get more and more of the organic acids. They have the lowest toxicity. So in the source area, you have hydrocarbons, so that they're really driving the toxicity, the risk. And down gradient, where you have just these polar compounds or these oxygen-containing compounds, we think the screening limits should be higher because the compounds are less toxic. And we summarized all this. We had a number of papers before the Zemo et al. 2007, but in that paper, we sort of summarized kind of all this GCGC work, the toxicity screening approach, and came up with these various stages that are typical for hydrocarbon plumes, and ways that regulators may want to develop screening levels based on those. And so, again, that's a good paper to read, and I can give you a resource for that. Or it's actually open access, so I can maybe through Dimitri, I can send folks a link. And different regulator, I don't have what we suggest here because different regulators have come up and used different final screening values based on this approach. Doing all this, we are looking at samples up gradient, down gradient, and within the source. And what we realized that the compounds that we see most regularly, we saw them in background samples and down gradient samples, which sort of suggests that these aren't unique to hydrocarbon degradation, and they're more, they're indicative of biological activity. Groundwater has bacteria, especially in the kind of living on the soils, and they're producing these chemicals as part of their normal process, and they get cycled through between the bacteria and the biomass in the dissolved phase as bacteria die and get eaten by predators and things. So then we sort of broadened our question to which are the compounds we're looking at degradation intermediates, which is what we sort of always thought, or some of them microbial products. And I define microbial products as the bacteria has basically totally degraded the compound hydrocarbon down to little pieces that they then build back up to grow. And so again, it didn't matter what they ate, the biomass, the chemicals are the same. So this is kind of the focus of our question. And what we did then is we started working with the USGS. They've been doing research at this crude oil release site in Bemidji, Minnesota for 30 years. So it was really nice working with the USGS. They understand this chemistry of the site, the geochemistry, the microbiology, and they started using one type of high-res mass spec. The Chevron folks used another. We are isolating samples a little differently, but working together, we came up with a new understanding of these complex chemical mixtures. And one thing going from GCGC to the high-res mass spec, again, we weren't limited by compounds that could get through the GC. So we're looking at more compounds to begin with, plus we have an analytical method that allows us to characterize, at least at the formulae level, more compounds. And so we're really looking at everything that is part of the dissolved organic carbon. And what's helpful in this is there's a whole field of research of people looking at dissolved organic carbon in mainly the ocean, but also other surface waters, because it's such a huge sink for carbon in the ecosystem. So there's a lot of climate change money. People are getting high-res mass spec, looking at how DOC changes over time in different environments. So we sort of brought that field of research into what we were doing. So this is just from what we have here is looking at 6 samples that we collected inside the plume. So down gradient of the source, or potentially where we still had hydrocarbons. But then we looked at what we see in the blue. Those are chemicals that we see down gradient, but those same formulae are also up gradient. The black are unique to only down gradient samples. So what this suggests is, again, a lot of the DOC concentrations is much higher down gradient, but the classes of compounds, the chemistry is similar. And again, we see not many, just a small percentage of the samples. Again, the black is formulae that are unique only to down gradient samples. There's a lot of, again, we get a lot of data, and there's just how to deal with it, excuse me, is always an issue. The one thing we look at is the chemical complexity, and we use a thing called double bonds equivalence, and I don't have time, or we don't need to go into exactly what that is, but basically compounds with higher double bond equivalence are more complex, they have more aromatic structure. Compounds with low DBEs are more linear, simpler. This oil, is very alkane rich, and the USGS has demonstrated that most of the degradation is going on is the degradation of alkanes. And it makes sense when we took the actual oil and looked at what we saw there. Most of the oxygen-containing compounds had low DBEs, indicative of alkane degradation. So right with the oil, what we saw looked like metabolites. But as we got down gradient, what we see here, this is samples of what's the organic oxygen-containing compounds in the source area groundwater. We see very high DBEs. It's much more complex than what's being degraded, and it's fairly similar to what we see up gradient. So, and there again, there's a lot of research on dissolved organic carbon in various environments, and what we're seeing in these down gradient samples is similar to what others are seeing in uncontaminated dissolved organic carbon. So again, suggesting that what we're seeing is more likely biologically produced compounds that are being recycled as bacteria die and are consumed by microbial predators. And so less likely, we can't say that there aren't compounds in this mixture that aren't just the hydrocarbons with some oxygen on it, but we don't think a very high fraction. Again, I think that's important in terms of the risk. If this DOC looks like DOC everywhere, you have a different sort of risk thought about it than if this is material that is really just hydrocarbons with a step of, just an oxygen or two added. And then just sort of putting our work into the understanding of DOC transformation. This DOC transformation continuum there's actually another paper that just came out in ES&T yesterday. People are looking at quite a bit in the ocean in terms of how DOC changes as it's degraded. And I think we're seeing the same thing. Samples that have high bioactivity, have high DBEs, and then it decreases as we go down gradient. For the microbiologist among you, this is sort of my conceptual model that most of the degradation is occurring right at the oil-water interface. This is a very fermentation, methanogenic driven process. You have a lot of methanogens and fermenters right at the oil-water interface. They're growing, producing a lot of DOC. And again, there's complete degradation of the hydrocarbons, but high DOC produced. I'm sorry, this has been on automatic, but here we are. So that's that case study. And again, happy to answer questions either at the end of the presentation or follow up with me offline. But I want to briefly talk about this new case. So this is the first paper on this was published early last year, and it's kind of been exploding. The researchers I'll have their names later at the end so you can contact them directly for information. But it's the Center for Urban Waters. It's sort of a collaboration between UW-Tacoma, Washington State, Puyallup. They've been trying to understand this salmon urban runoff mortality syndrome. Why are returning coho dying after rainfall in some of our urban creeks. And they've been focusing on, this important question for a long time. The chemists involved have gotten, there's some really good non-targeted analysis chemists involved in the effort. And with its collaborative effort of toxicologists and chemists, they came up and identified this 6-ppd quinone, and I'll show you what that is in a slide or two, as the most likely toxic agent. And looking back at what they think the 6-ppd quinone is something that's an oxidized product of 6-ppd, and it's an antioxidant that is added to tires. So really, they add quite a bit of this material with the whole goal of letting it oxidize so the rubber doesn't get oxidized by ozone in the environment, and then it washes into stormwater. And this is sort of a summary of how they did it. On the right, they did sort of a toxicity identification evaluation. They kept separating, well, they started with tire particles, made a concentrated solution, and then just kept running it through different columns, finding the fractions that were still toxic, looking at their chemistry, running it through a different column, continuing to isolate a smaller and smaller number of compounds, but the ones that are still toxic, and through that, and using their non-targeted method, came up with the identification of these 6-ppd and its family. And at the same time, on the left, they were looking at what compounds are in water. They had sort of a citizen science group that if a storm happens during salmon migration times, people go out, they look for, monitor salmon health, they look for dead fish, they take water samples, they send them to the lab, and it sort of gives an idea of, what chemicals do they see in these surface waters when they're seeing salmon dying. So it's a very kind of broad collaborative effect that they're doing. And again, this is just the 6-ppd, it's this nitrogen compound. It gets oxidized by ozone to form this quinone. And, again, that's the compound that they've proposed as something that's very toxic to fish. And it's in a lot of things. The tires themselves, I think the tire wear particles are the little pieces that are on the road that actually get into stormwater. There's the whole industry that use crumb rubber, sort of recycled tires for turf. They're concerned about this issue as well. And in some places, recycled tires are used in road mix. And the media that people are looking at is really stormwater and creeks, and beginning to look more and more at sediments. Again, this just sort of started last year, but DOEs, they're very interested. They've already had a stormwater working group, and it now has a sub-team focused specifically on this. They were hiring, and they may have already hired somebody whose really job is to follow this issue full time. California Department of Toxic Substance Control, they're doing it as their sort of environmental chemistry process. That's kind of the document they put out there on the right. Of course, the tribes are very interested in anything that have to do with fish. They have a lot of good scientists who have been very active. Oregon's getting involved. There's some in the sort of this Puget Sound SOR, local congresspersons are trying to get funding to deal with it, and NGOs have gotten involved. So there's a lot going on, and it's growing quick. And just to sort of show how fast this has caught on, Tien's paper was early last year, and just looking over the last couple of days, I found there's 30 plus articles published in the US, Australia, Europe, already looking at this issue. And commercial labs what they're trying to do is, it's been found with non-targeted analysis, but they're looking to they've come up with some targeted analysis, which again will make it easier. And that's sort of something this whole thing on non-targeted, it's kind of a mix of research, or mainly research now. I think it's going to become more useful in sort of less researchy things, but if particular compounds are found through non-targeted analysis, you can then switch over to targeted analysis to make it easy to follow those particular things. And just this is my final slide, a repeat of what I said earlier. I think what these technologies are going to do is we're going to find that there's contaminants or unexpected chemicals in more things than we thought. It's going to lead to the identification of new COCs. It gives us some better ways of evaluating the fate and transformation processes. Again, I like forensics. I think we're going to be able to use that as well. And so it's sort of a rapidly changing field. And I know this is sort of a high level presentation, but I just really wanted to introduce these concepts. And with that, I just wanted to thank for the 2 case studies. Again, Chevron Energy Technology Company, they're just switched their name to Chevron Technology Center, but they're chemists and toxicologists and hydrogeologists were very active and really drove the first case study. I want to thank the USGS for allowing us to collaborate at the Bemidji site, and Dr. Kodowski and McGuire from the Center for Urban Waters. They're the 2 PIs that really brought that other issue that I'm sure would be very interested to talk about it with you if you give them a call. And with that, I'll open it to questions and maybe hand it back to you, Dimitri, as our Masters of Ceremonies.
Thank you very much, Kirk, for the very, very interesting presentation. Does anybody have any questions for Kirk? I would like to get the ball rolling, certainly. I could ask you many questions about both the chemistry side and the analytical side of the things that you talked to us about today, Kirk. But let's keep it a bit more practical. Do you see? So you talked about these methods. It was one of the earlier slides, and you mentioned that there is a lack of a certification for non-targeted analysis methods. Do you see these methods being more standardized in sites and in regulation in general?
Probably, but maybe not for a while. I think it's going to be more project managers. Well, either people, who are interested in what's the contamination at a site, whether it's the person responsible for the site, an NGO, the regulators. They may say, well, we now know these things are here, and we're going to use them, and then everybody involved is going to have to respond to them. I think in terms of that, the EPA leading that effort to really understand, which of these methods work best, which ones are the most consistent, and trying to validate the different approaches. I think maybe when it's NTAC, I think it's the EPA's initial for their non-targeted effort, but I think when that's further along, then it may get to the point of being more standardized or certified. But I think the higher end commercial labs are going to provide this service. They see it as something that people are interested in, because I've always felt, I guess coming in from sort of researcher mode, even that, there's the analytical methods that regulators want us to do for certain things, and they're good for what they're for, but then we try to jerry rig those results into other issues. And I think often if you're a project manager at a site, if you understand what questions are that chemistry may help, it can be worthwhile to do your own chemistry, even if it's not a certified method, to help you understand your site.
Exploration, exactly. How big of a role do you expect cases like the one with the coho salmon mortality that broke out here in our neck of the woods?
Do I expect to find other unexpected?
Do you expect that they're going to have in order to get more acceptance, you need more breakout cases like that in order to, to showcase the usefulness of non-target analysis?
Yes, I think this is going to, I think this is going to become a high profile, sort of already becoming high profile, and with that how did we get here piece of it is going to come with it as well. And I think the other area where non-targeted is when it gets started to be used along with things that are already hot topics. I didn't talk about PFAS today, and I think PFAS analytical is, you could definitely talk an hour or more on just that. But on things like PFAS or PCBs or PAHs, where you have a lot of compounds in that family, I see the benefit of non-targeted, because again, you can kind of look at them all at once, as opposed to, well, we've always looked at these certain ones. And thinking about PAHs, we do a lot of forensics with that. And if you're just understanding risk, people do 16 priority pollutants. We do forensics. We're looking to do, maybe 30 or 50 PAHs. But if, again, often two similar sources, the compounds we look at look similar. But if we use broader methods, we can find, what's that one or two compounds that's in source A, but it's not in source B. And that is sort of the strength of our forensic approaches. Yeah.
Exactly. Anybody else have a question for Kirk?
If you didn't get my contact information, Dimitri knows how to get hold of me. So if you send him an email, we'll make sure that I'll, or I can, feel free to contact me. I like to talk about this stuff.
Absolutely, yeah. And if anybody has any question, when you see the video as well in the end about some of the papers that Kirk talked about during the talk, feel free to reach out to, request details about it.
Good. Well, thanks for giving me the opportunity to speak. And
absolutely, thank you. Thank you very much for the presentation, Kirk. And you're thanked by Jessica, Stephen.
Thanks.
So, thank you very much, everybody, for attending as well. And we're 15 minutes short, but feel free.
I figure people feel free. And I will, I'll put together, I didn't get time for the reference list, but I'll put this little bibliography with links, and I'll send it to Dimitri, and maybe you can send it out to your mailing list.
And share it out, absolutely.
Okay, thanks everyone.
Yes, thank you very much, everybody. See you next month again.